We've got our identification notes on microscopic cells and organisms. You're going to find a great deal of resources here with mammalian red blood cells, avian blood cells, a whole bunch of different things going on. This is from IDEXX Laboratories. It's actually your comparison between canine and feline red and white blood cells, so if you wanted to take a look at those. eClinPath is a great resource for you when you are looking at different cells and cytologies. So that's going to be a great resource for you guys to take a look at. So we are going to go ahead. Our notes should be set up, our names of our organisms or cells, your drawing and characteristics of that cells or organism. Then we've got the first thing we're going to look at is our epithelial cells or our epithelial cells. Our epithelial cells are pretty much our skin cells, right? So they are going to be the skin cells that cover both the outside and the internal surfaces of our organs. So pretty much that first layer of our organs and our body are going to be epithelial cells. So we'll find these sloughed off in ear cytologies, we'll find them sloughed in urine analyses, and so we'll do skin scrapings, right? And then we'll really see them because they'll be in the skin scraping. So this is just an example of the epithelial cells found in urine. So these particular ones just happen to be round, and then we have that little nuclei there of the cell. Then here's some epithelial cells found in the ear. These are actually from Hutch's ear. So you can see there's the folds in them. They're not necessarily uniform in shape because when you think about it, skin almost, I like to think of paper, right, when I'm thinking about epithelial cells. So it almost can be crumbled up. Next thing we're going to look at is a bacteria called coccibacteria or cocci. That is going to describe our spherically shaped bacteria. So cocci are in a sphere. Remember, everything we look at underneath a microscope is actually three-dimensional. We're looking at it in between a layer of two pieces of glass in liquid. So there are three dimensions to our things. Here's our cocci. These are cocci that happen to be all clumped together, but you can see the circles. And this is at a very high magnification. They were also stained, so we could see them more clearly. The next type of bacteria we're going to look at is bacillus. So I think of bacillus with my parallel L's. They are going to be rod-shaped. So where cocci are all circles, right? And that's the shape that they are. With bacillus, we've got the rods of the L's. So they're rod-shaped bacteria. And so here we have those lots of little rods. Okay, so little tick marks, little hashes. So that's our bacillus. And then this is a different, these are stained with what we call gram stains because we can tell like the grouping of the bacteria. We call them gram positive or gram negative. We'll learn more about that though as we go. Right now, I just want you to think about those shapes of the bacteria. Next, we have a fungus, which is yeast. So yeast is pretty interesting. There's over 1,500 different species of yeast. It is a single-celled fungi. There are non-pathogenic species, right, which we use to assist in gastrointestinal processes. So we might put them as an additive. We might use it as a probiotic or prebiotic. We also use non-pathogenic species of yeast in the food and beverage productions and industries. Pathogenic species, however, are often going to flare as a result of a stress or compromised immune system. So we'll often find that it's what we would call a secondary infection because, so with our rabbits, maybe it was allergies, right? So they had allergies, which then lowers their immune system, which causes the yeast to flare up. So those things, you know, because I have a weakened immune system, I can't fight off my own yeast, so then therefore it flares. So I just think that's the niftiest thing, like 1,500 different species. And again, not all yeast are bad, right? Because we use them to bake bread and other things. Next, we're gonna, so our actual yeast, So to see what it looks like, we're just looking at the pathogenic organism. So these are in the lagomorph ear cytology, right? So this is Hutch's ear cytology. So we have those really dark purple dots right here. Now, if bacteria were in this slide, it'd be even smaller. So that's how we were able to differentiate and know that that's yeast and not bacterium. Next, we're going to go into our blood. So neutrophils are a type of blood cell that's only found exclusively for mammals. So neutrophils are going to be a type of white blood cell that is going to engulf our invading microbes. We generally have the most neutrophils. We should have the most of in our body because they're like the frontline infantry. And so here is what a neutrophil actually looks like, which we have, these are banded and segmented neutrophils right here. So we have, that's the nuclei, right? So that really dark part is the nucleus. And then you have the outer the outer cell membrane, and the speckling in there, that is the cytoplasm. So this is a granulated cell, which again, we'll get more into when we talk about cells next semester. And then we're going to look at mammalian thrombocytes. Neutrophils, I should make sure I specify, neutrophils are only found in mammals. So in other organisms, in your fishes, in reptiles, birds, amphibians, you're going to have heterophils. So they won't be neutrophils, they'll be heterophils. So they're just slightly different. So going into thrombocytes, we're going to specifically look at mammalian thrombocytes, because as we go between different classifications of animals, we do find that that cellular structure is going to change, right? We're not all the same. All the animals, they all are different. And even between different species of mammals, you'll see some differences. But these are kind of your general guidelines. So mammalian thrombocytes, or platelets, which thrombocytes are platelets, are going to help us form blood clots and slow or stop the bleeding, right, and help our wounds to heal. So they're the very first line to be able to start plugging those holes when we get a wound. So here is what our mammalian thrombocytes look like. So it has that little cluster, that dark red nuclei, and then it starts to branch off. So this branching is what's going to form a cyst in like attaching and making those blocks inside of your wounds. Then we have a mammalian erythrocyte. So erythrocytes are red blood cells. So with erythrocytes, it's pretty nifty. Mammals, our red blood cells, regardless of our animal species, are carrying oxygen, carbon dioxide, and other elements throughout the circulatory system. So we have nitrogen in our bloodstream. We have different other waste gases and products. And so the red blood cell is primarily transporting all the gases. We just often think about our oxygen because that's the only way our oxygen is getting throughout our body, right? So with mammalian blood cells, many of you have seen them, they look like donuts. So it's a spherical shape with a concave center. There's no nucleus. So with these guys, so this is a bovine blood smear, but you can see these perfect, just like little circles, right? And then they are empty in the inside, so they get really thin on the inside. Here's some of those platelets too, right? Boom, from far away. But so that is a mammalian blood smear. Boom. All of those red circles, empty sphericals, no nuclei present. Then we're going to talk about non-mammalian erythrocytes. So these are everything that's not a mammal that has blood. So still, again, responsible for carrying oxygen, carbon dioxide, waste gases. But these are going to be ovular in shape, and they actually have a nucleus. So there is a nuclei present with these. So here's avian erythrocytes. So we see that oval, and then we see that nuclei inside. Okay. So that's our nucleus, that's our oval. Then we have reptiles and amphibians, or herptiles, which is where we get herpes from. Not herpes, totally different. But so if you want to be a herpetologist, this is where that comes from, so herptiles. So this is their red blood cells, and here's a few different species of both reptiles and amphibians. So you can see the different gradients of the ovular shapes. And then there's some, so these would be some white blood cells. Again, they're named differently, and they look different between mammals and non-mammals, which is why we're not going into the difference of all of those. It's beyond the vet assistant's pay grade, right? You don't need to know those things. So those are reptiles or herptiles. And then we have fishes. So our fishes are pretty cool too. Again, still ovular. Some of them are a little bit more circular. These are all just different species of fish. So you can see, again, they're all ovular, and they have that nucleus. So when in doubt, like, it's got a center, right? So there's definitely a center present. All right, and so those are all of the terms for us for this identification quiz.